take two cannabis flowers with the exact same THC percentage, same number on the label, now smell them. One sharp and the citrusy, the other earthy and musky. And people will tell you they will feel completely different too. So if the THC number is identical, what's actually different? That question is what today is about. Because last episode we tore down the sativa versus indica labels, and I promise you the system scientists actually use instead. This is it, and it has two dimensions: what the plant makes in terms of cannabinoids and what it makes in terms of aroma compounds, chemotypes and the terpenes. Quick note before we start: this is plant science, not a medical advice, and I will be careful to flag where the popular claims run ahead of the evidence, because in this topic they do. So let's start with the cannabinoids. Instead of a vibe like indica, scientists thought cannabis wide chemotype. Literally, it's chemical type. What does this specific plant actually produce? And it turns out there are 5 clean categories. Type 1 is THC dominant. Lots of THC, very little CBD. This is what most people picture as drug cannabis. Type 2 is balanced. Meaningful amount of both THC and CBD, roughly in the same ballpark. Type 3 is CBD dominant. Lots of CBD, very little THC. A lot of medical and hemp cultivators sit here. Type 4 is CBG dominant. That's the mother cannabinoid from episode 1, showing up as the main product instead of being converted onward. It's rare. And type 5 is the strange one, especially no cannabinoids at all. These are pure 5A plants. Now here's the beautiful part, and it connects straight back to episode 1. Remember the enzymes THCA synthase and CBDA synthase competing for the same raw material CBGA. Whether a plant is type 1, 2 or 3 comes down to which of these enzyme genes is carrying and whether they work. It's controlled at especially a single genetic location, and it behaves like a clean, predictable inherited trait. That's why chemotype is so useful. It's measurable, it's stable, and it's genetic. This is not new either. Chemists were classifying cannabis. This was way in the 1970s, long before the marketing label took over. So that's dimension 1. But it doesn't explore our two flowers with identical THC. For that we need the second dimension. So that dimension 2 is terpenes. Terpenes are the aroma compounds. They are what you actually smelling when you smell cannabis, and they are not unique to cannabis at all. They are the same class of molecule that makes pine smell like pine and lemon smell like lemon, and they come off the same assembly line we drew in episode 1. So remember one of the two raw material stream feeding into CBGA? The same precursor GPP is the starting point for terpenes too. So the plant is building its cannabinoids and its aromas from overlapping chemistry in the same little resin glands. Let me give you the main players because their names double as their smells. Myrcene, earthy, musky, herbal, is the most common one in cannabis. Limonene, citrus, exactly what it sounds like. Pinene, fresh pine. Linalool, floral. The lavender note. Terpenol, bright and complex. Beta-caryophyllene, black pepper, spicy. And here's a lovely callback. One of them is called humulene, named after Humulus, hops, the beer plant that shares an ancestor with cannabis from episode 1. The family resemble is literally in the aroma. And these are made by their own set of genes, terpene synthase genes, which ties up a loose thread from last episode. Remember how the sativa versus indica labels were genetically almost meaningless, except for one small kernel. That kernel was terpenes. The labels weakly tracked variation in exactly these terpene synthesis gene. So, the tiny bit of truth buried in sativa and indica was never about the genome as a whole. It was about aroma. So now we arrive at the big idea everyone wants to talk about, the entourage effect. The claim is that cannabinoids and terpenes don't act in isolation, they work together. So the whole plant produces effects that pure THC or pure CBD would not. It is the reason people say two flowers with identical THC can feel different. And here's where I have to be straight with you, because this is exactly the kind of claim that get oversold. Is it possible? Yes, there are genuinely interesting mechanism hints. For example, one of those terpenes, beta carotene, actually binds to one of the body's own cannabinoid receptors, which is a real pharmacological interaction, not hand waving. And there's a growing body of early research, including some preliminary clinical work looking at whether specific terpene change how THC feels. But, and this is the important but, the honest state of the evidence is that most of it is clinical, meaning cell studies and animal studies, and the idea that terpenes meaningfully boost cannabinoid effect in actual patients remains unproven. A recent scientific review looked hard at this and concluded essentially that the synergy is reasonable hypothesis but not yet demonstrated, and that we need proper clinical trial to know. So the entourage effect is not a myth like sativa versus indica, but it is not established fact either. It is sits in that honest middle, promising, mechanically possible, and genuinely unsettled. Anyone stating it as proven is getting ahead of the science. So, put the two dimensions together, the cannabinoid chemotype and the terpenes profile, and you get something far more useful than a strain name. You get a chemical fingerprint. Researchers call this the shift from cultivator to chemovar, identifying a plant not by whimsical name or a vibe, but its actual measured profile of cannabinoids and terpenes. And for anyone working in medicines rather than the consumer market, that's the whole point. If you want a product that consistent, the same thing batch after batch, that's the clinical and a patient can rely on. So you cannot define it by indica or by a catchy name. You define it by its verified chemical profile confirmed by lab testing. Chemotype and the terpene fingerprint are the language of standardization and reproducibility. That's what makes a plant into a medicine grade product rather than a lottery. So it brings it home. Chemotypes types 1 through 5 tell you what cannabinoids a plant makes, and it's genetically clean and predictable. Terpenes add a whole second dimension of aroma and possible more, made by their own genes, and they are the real kernel of truth hiding inside the all sativa indica labels. The entourage effect, the idea they all work together, is plausible and interesting, but honestly is still unprovable, and the future belongs to the Chemovar, describing cannabis by its chemical fingerprint instead of its name. Next time we go inside the body, the endocannabinoid system, and how these compounds actually interact with us in the first place. That's episode 3. Thanks for watching.